And today I'm going to be presenting, I guess, one part of an argument that I've been making, trying to come up with a definition of the organism. So this is a little bit of a change of pace from the previous talks, getting into a little bit more of the definitional philosophy around the concept of the organism. So to spoil the ending today, I'm going to be making kind of a bold claim, and that is to work around how we actually think about the organism concept. So the first part of this is that the organism concept should be capable of describing some sort of biological entity or specific sorts of biological entities, enabling us to distinguish organisms from other sorts of biological entities, such as parts of organisms or ecosystems in which they reside. And to do this, I'm going to argue that we should adopt a materially restricted concept of the organism as having what I call a centered metabolic network. So this discussion fits broadly into the topic of the problem of biological individuality. And I see the problem of biological individuality as the search for the criteria that underlie the concept biological individual. When thinking about the problem of biological individuality, I endorse what I call a principled pluralist approach to the problem, which recognizes multiple kinds underneath the umbrella concept biological individual. And most of the literature that has this kind of pluralism tends to focus on two distinct kinds of biological individuals. This includes evolutionary individuals or Darwinian individuals and organisms or physiological organisms. Evolutionary individuals are those entities that are capable of forming lineages. They are the entities seen by natural selection and are members of an evolutionary population which fulfills Lewontin's criteria of having variation in traits. These traits are heritable, and these traits lead to differential fitness between individuals in the population. In contrast, physiological organisms are defined by physiological criteria. In this, they can have any kind of reproductive capacity and any kind of evolutionary history. They are self-maintaining entities, and they're ontogenetic or developmental, and they often represent the cooperation between diverse parts in a physiological sense. So the way we can conceptualize this is that the term biological individual is an umbrella term which refers to either or evolutionary individuals or organisms. So while many entities will be both an evolutionary individual and an organism, these two concepts can come apart. So while our mouse here fulfills both the criteria of being a reproducing whole and a physiological whole, our virus is only a reproducing whole. It does not have the physiological mechanisms to make it an organism. And likewise, on certain views of the evolutionary individual, particularly those that emphasize a reproductive capacity, the mule, a sterile hybrid, does not have the capacity to reproduce and is therefore not an evolutionary individual, but would be an organism. Importantly, I don't see these criteria as strictly categorical. Rather, it's better to think of them as coming by degrees. So we will have some entities that fulfill both sets of criteria to a very high degree, and many entities that kind of sit in this liminal space. I'll use this instead. In this kind of liminal space here, where it's a little bit unclear whether they're either an evolutionary individual or an organism or neither. I will say that in another paper, I do argue for a third kind of biological individual, which is guided by goal-directedness or purposiveness, but I'll leave that to the side for now, and we could maybe talk about that in the discussion. So the problem of biological individuality is often discussed when turning to problem cases that are hard to categorize. So these cases like the virus or like the mule. And one of my favorite cases to discuss is the case of the eusocial honeybee Apis mellifera. And this is because we often ask the question, is the honeybee hive itself a biological individual? And this is because of it being a eusocial insect. So eusocial colonies like bees or ants or termites, or not termites, they have a different structure, but bees or ants have one female queen that does all the reproducing, 20,000 to 40,000 sterile female workers, and at certain times of the year, 200 to 400 male drones. Now, there is a very distinct division of reproductive labour in the hive, as well as a distinct non-reproductive division of labour, as in the worker caste. So if we ask the question, is the honeybee hive a biological individual, the it's fairly, I think, straightforward to say that it is an evolutionary individual. Reproduction is occurring at the level of the hive and not at the level of the single bees within the hive. However, it's much less clear whether we should consider the honeybee hive an organism. And this is because of a divide I see in the current literature of solutions around concepts of the physiological organism. So those solutions which I call abstract solutions that lean entirely on kind of more conceptual notions like self-maintenance, integration, cooperation, tend to say yes, the honeybee hive is an organism in its own right. It's self-maintaining, it's cooperative, it's integrated. However, more specific solutions such as spatial boundedness, metabolism, or immune tolerance in a strict biochemical sense would say no, the honeybee hive isn't the organism, but the bee is. So one common solution to the problem of the organism has been proposed by Queller and Strassman, where they define the organism as the largest unit of near-unanimous design and as a unit with high cooperation and very low conflict among its parts. So they define the organism as being a cooperative entity that can be realized in multiple different ways. And according to their definition, yes, of course, the honeybee hive would be an organism in its own right. And in fact, the authors do state this in their paper. However, there is a problem with their account. Queller and Strassman emphasize that the organism needs to be the largest unit of near-unanimous design, effectively including what Godfrey Smith calls an exclusion principle, where an organism cannot be a functional part of another organism, at least not to the same degree. So this doesn't have to be strictly categorical. Rather, even though the honeybee would be displaying organismal properties or organismal traits, it loses organism points by being subsumed into the larger hive organism. So what this means is that it needs to be either the bee or the hive that is the organism. It can't be both. So if we say that the honeybee hive is an organism, then the honeybee cannot be. So I think there is something wrong with this conclusion, that the hive is the organism and the bee is not. And this comes down to how we often use the organism concept in the biological sciences. So let's compare two bees. So we have our European honeybee, Apis mellifera, and a solitary Australian bee, the blue-banded bee here. So our solitary bee, unlike the social bee, is capable of reproducing on her own. She nests, forages, and does everything on her own. On any definition of the organism, our solitary bee would be an organism in its own right. There is no question about this. However, on these more abstract solutions like Queller and Strassman's, our social bee is not an organism. Now, while these two bees have very different reproductive strategies, they're physiologically comparable. They're often compared, and they have pretty much the same anatomical physiological structures apart from those related to reproduction. So it seems a bit strange to me, at least, that we would say that the blue-banded bee is an organism and our honeybee is not. So one solution would be just to throw out the exclusion principle and say that we can have organisms the whole way down. So an amended version of Queller and Strassman's account would be that an organism is a unit of diverse parts with high cooperation and low conflict, where adaptations of the parts do not disrupt the whole. So in this case, we would say, okay, yes, the honeybee colony is an organism, and so is the bee. Fantastic, we've solved the problem. However, unfortunately, so too would its cell, and so too would the mitochondria. So we lose the power of the organism concept as picking out a distinct level of organization in biology. An alternative solution is to adopt a materially restricted concept of the organism, where the organism represents a particular sort of biological entity which is both countable and identifiable over time. So rather than solely being defined by abstract notions like cooperation or integration or even self-maintenance, we should look at how these concepts are grounded in real biological entities and go from there. So this would be much like how we think of other kinds of biological entities, at least their operational concepts. So for example, the gene, although it can be broadly defined as a unit of in the Mendelian sense as a unit of inheritance, in operational definitions of the gene, it tends to at least be thought of as a piece of nucleic acid or be made of nucleic acid, and often is also defined by having a functional output such as producing an RNA or a protein, or some relational condition of being part of a gene regulatory network. So a similarly materially restricted organism concept would provide the same kind of operational grounding for counting and identifying organisms at a particular level of organisation. So the question is, what are the material conditions that are necessary for something being an organism? So to quote Goodwin and Dawkins, biology is the study of life, and life comes in the form of organisms. So I see this feature of it being a living cohesive whole capable of self-maintenance which persists over time as being a defining feature of organismality, and this is how we can distinguish a living organism from other kinds of self-maintaining entities like nation-states, social groups, or institutions. So what is life? So despite some much disagreement about definitions of life, generally definitions of life invoke metabolism as a feature. To be alive is to metabolize, and metabolism in a broad sense is the conversion of energy from the environment into usable energy that allows the living entity to persist over time. It keeps it out of entropic equilibrium with its environment. Metabolism is not mere chemical transformation of energy, but transformation for a particular purpose, and it can include the creation of biomass or the creation of energy by breaking down complex molecules. So metabolic accounts of the organism are not new, there's plenty of them. However, all of them tend to emphasize that metabolism alone is not enough to define the organism. So the organism tends to be thought of as a metabolically self-sustaining whole, or a system of diverse parts which work together to maintain the system structure. It is functional integration through metabolic collaboration. So this idea of collaboration, of integration, is invoked when we talk about the metabolic organism. But there's a question. What makes something an integrated, self-sustaining metabolic whole? A feature of metabolism is that metabolic reactions don't occur on their own. They're not just kind of mere chemistry, but form these multi-step pathways which then form these complex metabolic networks. A metabolic network is defined by certain metabolites from one pathway becoming reactants in another pathway. And in eukaryotes, and these metabolic networks are compartmentalized across the system, both within the cell and between cells, enabling metabolism to happen at this whole network level. So maybe we can just say that the organism is a metabolic network. However, being a metabolic network alone is not enough to define the organism. In a broad sense, if we were to define a metabolic network as being the reactants of one metabolic pathway or the products of one metabolic pathway forming the reactants in another metabolic pathway, then ecosystem level interactions would count as metabolic networks. So taking a very kind of simplified version of an ecological interaction between our tree and our bird, we see that the results of respiration of the bird become the reactant in photosynthesis for the tree, and on it goes. Their metabolic networks are shared. So between organism interactions could be considered a kind of metabolic network, so we lose that ability to demarcate the organism from its environment. So unless we want to fully embrace the Gaia hypothesis and say that the whole biosphere is an organism, then we need some account of the difference between the chemical transformations that occur within an organism and those that occur between organisms. And I think here we can turn back to this idea of self-maintenance and look for the features of a metabolic network within an organism that enable the kind of self-maintenance we see within organism metabolism that we don't see at the ecosystem level. So rather than the organism merely having a metabolic network, the way metabolism is organized within a whole organism is that it's centered. It's centered and directional and coordinated in such a way that enables the system to be robust. And so they must have function and be structured in such a way to maintain this robust flexibility necessary for organismal level metabolism. So the first place I looked when thinking about this idea of centeredness was in work being done in metabolomics. And one thing that became apparent is that organismal metabolic networks across all three domains of life have this bow tie structure, where a large number of nutrients are transformed into a very small number of reactants or metabolic intermediaries, which then become a large number of products. And this core metabolism seems to be conserved across all domains of life. So Mann and Zeng in 2003, starting there, found this kind of metabolic network topology and with this highly conserved metabolic core. So what seems to be the case, and what has been postulated by researchers in this area, is that this kind of bow tie structure is incredibly robust to environmental changes. And as Ma and team state, despite the diversity of possible biochemical networks, it may be common to find that only a finite set of core topologies can execute a particular function. These design rules provide a framework for functionally classifying complex natural networks. So what they postulate is that there's something kind of fundamental to this kind of network structure to create the kind of network needed for the kind of life we see in organisms. However, I hesitate to read too much into this data as a means to identify the organism. And this is for a few reasons. The first is that there's bow ties everywhere. So even if we look at this diagram by Seston Doyle, there are three distinct bow ties here. We have the main metabolic bow tie on the outside, we have the core metabolism also forming a bow tie, and gene transcription networks also form bow ties. The internet's also a bow tie in a network structure. But the second two are a bit more concerning. So in all of these studies, organismality is already assumed based on a genome model of the organism, so using things like the KEGG database. So it would be circular to define the organism from studies that already have defined the organism using a genome model. And because this is a genome-based model, apart from some of the model organisms there, a lot of these metabolic pathways have not been experimentally confirmed. So it's not really clear that these kind of genome-based analyses are giving an accurate picture of how metabolic reactions are actually interacting in situ. However, one key feature from these investigations I want to focus on when considering these network topologies is what seems to be consistently showing up across all these studies is that there are these core intermediaries of metabolism that the wide variety of nutrients are always transformed into, and which the metabolic network seem to be structured around creating. These metabolites, so Ito and team have said that in the metabolic networks, various nutrients are decomposed into 12 universal precursors, which are then used to generate abundant biomass. So these include metabolites like glucose-6-phosphate, pyruvate, acetyl-CoA, among others. And a key feature of these metabolites is that they're too environmentally unstable to be picked up from the environment by the organism, but they are highly necessary for the kind of metabolism that occurs within organisms. So the organism must be able to create these metabolites themselves. So I hypothesize that it is via the production of these unstable intermediary metabolites that a certain amount of robust flexibility is enabled within organismal metabolism. More importantly, because these metabolites are chemically unstable, the organism must be able to produce them themselves. So we'll be looking for metabolism that is centered around this production. Now, I don't want to say that the organism must produce specific metabolites. That would be much too narrow to say that something is an organism if it produces pyruvate. But rather, I think organismal level metabolism may be centered around the production of environmentally unavailable chemical intermediaries with specific biochemical features necessary for the kind of metabolism needed for life. So this idea of specific biochemical features has been also discussed in papers looking at questions of the origin of life. So Smith and Morowitz make the claim that core intermediaries in the reductive tricarboxylic acid cycle, or the reverse Krebs cycle, such as pyruvate acetate and alpha-ketoglutarate, represent a self-organizing anabolic core from which all other metabolic reactions are derived. And this metabolic core, they argue, is universal and it is primordial. They argue that it's one of the earliest forms of metabolism. Now, this is an empirical question for those who are experts in this area. However, one feature that they point out is that these metabolites have a very specific reactivity, and it's this feature of them being stable enough to remain in their cellular environments and to be available for multiple pathways, while also being reactive enough to be able to be the reactants in multiple pathways. Their reactivity is not so specific that they can only be doing one job. So one example of this, not in the reverse Krebs cycle, is acetyl-CoA. And Martin and Russell in 2006 emphasize this as being one of the most primitive biochemical pathways for carbon fixation or CO2 fixation. And the authors emphasize why this is the case based on acetyl-CoA's reactivity as a thioester. So they say that thioesters are a very rich class of compounds with high transfer potential to participate in other reactions. Accordingly, the energy in the thioester bond can be used for many kinds of metabolic reactions. So they emphasize this idea that there's a specific kind of reactivity that needs to be there for these chemicals to act in such a way that they can help regulate the metabolic system. So importantly, why I think this feature of these intermediaries is important is that they enable a kind of metabolic self-maintenance of a system without the need for any further control, so any extra kind of mechanisms to control the movement of metabolism. So let's take one of the intermediaries, glucose 6-phosphate. So glucose 6-phosphate is the first intermediary in the glycolysis pathway, and often will continue going down the glycolysis pathway, eventually being part of the ATP cycle. However, if all the enzymes are full, or there's a lot of glucose, then glucose 6-phosphate could also be diverted down the pentose phosphate pathway to become nucleic acids, or down the glycogen pathway to become storage. And so without any further control, we have this ability for metabolism to be self-maintaining and self-regulating, maintaining a consistent level of energy in the system. So I hypothesize that when we're looking for organismal level metabolism, we could use these intermediaries as a proxy to see whether the network is structured around this core. And this is different to the kind of metabolic networks we see in an ecosystem. So there's a second feature of these centered metabolic networks which I want to discuss, which is this idea of metabolizing as a whole. So one criticism of this first part is that, well, in a multicellular organism, you have most of this metabolism is occurring within the cells, and then there's just the sharing of kind of endpoint metabolites between cells, and that's much like an ecosystem anyway, so this doesn't tell us anything. However, this is not necessarily the case. So when we see metabolism at the whole level, there is this distinct metabolic boundedness and compartmentalization of metabolism, which leads to metabolic division of labor. So there is one feature which is external boundaries of the metabolic unit, which prevents energy from dissipating freely into the environment. There's kind of a block there. But then there's also internal metabolism, which can be in eukaryotes via membranes, or in prokaryotes via cytoplasmic gradients or metabolic metabolons, enzyme components. So this kind of compartmentalization can lead to the structuring of the metabolic network by dividing the metabolic division of labor across different parts of the organism as a whole. So Barpeld and Corey identify 3 pillars of compartmentalization for metabolism. The first is the establishment of unique chemical environments. So different key metabolic pathways require very specific chemical environments to continue. And these environments may be detrimental to other key metabolic pathways. So compartmentalization enables this division of metabolic labor. The second is protection from reactive metabolites. So many intermediary metabolites are highly reactive, even more reactive than the intermediaries, then I've pointed out. The key example being reactive oxygen species, which can not only damage other metabolic pathways, but also cellular machinery, including nucleic acid. So the compartmentalization of metabolism protects parts of the cell and other metabolic pathways from these reactive metabolites. And ultimately, this enables the regulation of metabolic pathways across the system through this kind of physical structuring of metabolism. The control of metabolism between compartments, both within and between cells, is often facilitated in more complex organisms via signal transduction pathways. And so the unique chemical environments enable acute sensing of various metabolic states, and which can cause signaling cascades that can alter not only metabolic output, but also gene transcription and subsequent enzyme production. So the compartmentalization of metabolism also, particularly via membranes, enables the creation of energetic gradients, which leads to the production of ATP. So without compartmentalization of metabolism, we wouldn't be able to generate energy. So these two features of core metabolism and compartmentalization have been postulated as being a feature of the origin of life. So Harrison et al. Under Nick Lane in 2003 published a paper showing how rudimentary carbon fixation plus the boundedness of metabolism in hydrothermal vents could lead to the spontaneous generation of nucleic acids. So it's these two features that might be the starting point for life. So one thing I quickly want to mention is that because of the graded nature of organismality and of life in general, we're likely to find entities where it's not clear whether they are fully organisms or are part of a different organismal whole. So for example, cross-feeding bacteria and biofilms are able to share their cytoplasm, thus sharing all of their metabolites. And so I think there'll be many entities where it's a little bit unclear, where they can kind of play a bit fast and loose with individuality. So prokaryotes can do this generally, not just with physiological or metabolic organismality, but also with evolutionary individuality, sharing genetic components through horizontal gene transfer. However, I still think this account gives us an operational way of measuring organismality. So taking 3 examples, we have 3 cells which are being organisms on their own. And so what we would see here is much more like the ecosystem case, where each have their own metabolic networks represented by this kind of orange hourglass, and they're sharing waste products only. So they are sharing metabolites, but they're not sharing these more unstable intermediaries because they don't have the mechanisms to. In the in-between cases, like with the biofilms, there'll be some sharing of the intermediaries, but maybe minimal metabolic division of labor. And this may not be entrenched. They may be able to come and go from these metabolic collaborations. And when we're getting to a metabolic organism that's highly entrenched, we would see that there is one metabolic network with deeply entrenched metabolic division of labor and sharing of intermediaries between metabolic compartments. Okay, putting all this together in the last minute. So I postulate that there's 2 features of a centered metabolic network. The first is a production of core intermediary metabolites, and this enables the self-maintenance of metabolism through the creation of environmentally unstable intermediaries, and that metabolism would be structured around these intermediaries. And metabolic compartmentalization and boundaries, so metabolic integration and maintenance of the system, are enabled through the creation of distinct metabolic environments and metabolic division of labor. And this enables the electrochemical gradients necessary for the transformation of energy. So going back to the case that started it all, if we're looking at our honeybee and our hive, although the honeybee hive does have a slightly more complex kind of broadly thought of metabolism with the sharing of food sources, but also the creation of complex food sources like royal jelly. The kind of energy conversion and metabolic network needed for metabolism necessary for life is occurring at the level of the bee, and therefore the bee is the organism and not the hive. So I propose a definition of the organism as a cooperative collection of biological parts that are unified by a centered metabolic network, self-maintaining and resisting entropy by turning energy from the environment into usable energy for the whole organism. They are metabolic wholes. Thank you.